In this video, we will talk about rubella virus. Let's talk about the rubella virus and understand its structures. So it is spherical in shape, which is enveloped, and this envelope is derived from the host membrane. So basically, the lipid component of the envelope is from host membrane, and it has glycoproteins, which are generally known as E1 and E2, and it helps the virus to getting into the host cell. Then there is nucleocapsid, which protect the genomic material, and it's a icosahedron capsid. Rubella is also known as German measles. Talking about genome, here the genome is basically a single-stranded RNA, which is positive sense and roughly 9.8 kb in overall size. Then about the classification, rubella virus fall under the genus Rubivirus and the family Togaviridae. Let's talk about the epidemiology. So, the reservoir of rubella virus is basically a human. So, human are the natural host. Transmission can occur via respiratory droplet, which is common for measles, which is common for mumps also. These all three infections can be transmitted via respiratory droplets, but in case of rubella, especially the infection can be transmitted vertically from the mother to the fetus. Rubella can cross the placental barrier and affect the fetus. The first trimester is more vulnerable for the rubella infection, and this is quite dangerous because it can cause congenital rubella. The incubation period is 14 to 21 days, like any other viral infection. And basically, there could be outbreak of rubella at any point of time. Now, replication of rubella happens inside of the nasopharynx and eventually in the regional lymph node. Rubella virus has E1 and E2 proteins, which dock with specific cell surface receptors and the nasopharyngeal epithelial cells. Since it's a positively stranded RNA and it has mRNA-like features like the cap and the poly A tail, it can directly be transported to ER, and it can be used for protein production. Anyway, the proteins that are produced are shipped via the Golgi apparatus and eventually on the surface of the cell. A new virus particle is assembled and then eventually released. So this is how the infection works. Let's talk about the pathogenesis in bit detail. So first the nasopharynx and lymph nodes are affected. Eventually the virus enters the bloodstream causing viremia. And basically it can affect several other tissues and organs including brain and liver. So basically immune response against this virus is by humoral immune response. That means antibody mediated response or via cell mediated response, which involve cytotoxic killer T cells. Let's talk about the clinical features. So in prodrome phase, there could be mild fever, swelling of lymph node, and eventually there could be rashes. These rashes can be occur in the face first and separate throughout the trunk and the other body extremities and last over 3 to 4 days. The diagnosis is depend on clinical history and basically clinical presentation. The rashes, basically the rubella antibodies, the IgMs can be detected in the blood of infected patient. The PCR is best diagnosis for any viral disease. When it comes to treatment, supportive treatment, hydration might help to control the things. Sometimes vitamin A is given, hospitalization might not be required or might be required in severe cases. The prevention is better than cure, so this particular infection can be prevented by MMR vaccine. Every baby should be given this MMR vaccine, and actually MMR vaccine has reduced the chances of infection several times. Thanks for watching my video. Stay safe.